We did bring Raymond Brugada in. He's known for Brugada syndrome. Ralph Amatulum discovered THC and has done a lot of the work, the cardiac work up there with THC. And Lester Grinspoon, who's local and knows quite a bit about this field, and a few cardiologists in the Agincourt team. But this really set us on a mission to re-look at how do we get quality and safety testing, not just into epilepsy genetics, but on the plant that people are using. Because it was pretty clear from our experience that there's a lot of people selling snake oil right now, calling it hemp oil, because there's such a high demand for people to use this in epilepsy. And the epileptic kids, if you go back to the slide here, he was using 25 mgs 3 times a day. He's about a 75 kilogram person. That's about a 1 micromolar dose. It's very low. The actual epileptic children are using 5 times that amount per their weight. I mean, they're really jacking this up to 300 to 600 milligrams for these patients. And so this is a very subtle amount, but enough to be pharmaceutically active from what we've heard. But even that small amount's hard to find because the epilepsy patients are needing so much of this. So this got us rethinking about how to bring more safety and quality testing into the cannabis field. And this led us back to some of the work we did before. Many people have seen this slide, but this is the reason why we want to bring this into a fast-growing cannabis field, is that this is one of the most explosive technologies that we can think of that's going on today. If we can bring next-generation sequencing into one of the fastest-growing markets in the United States, there's bound to be some opportunity there. And you'll notice something happened around 2010 and 2011 when NEB hired a bunch of technicians out of LifeTech. The price stopped changing, and that's all their fault. What we've done is the first thing we want to do is we need a peer-to-peer system to share DNA throughout the field. We cannot move plant matter over state lines, even though it's federally illegal, but it's recreationally legal in 5 states now. And even up in Portland, Maine, I hear they went recreational. So DNA, we believe, can go across state lines as long as there's no THC in it. And if we can get DNA preps out there that can enable that, we can get people to start sharing genetics, and that would be a real help in the field. The other thing that they have been mandating, and probably for good reason, there aren't a lot of recorded deaths on people using cannabis. The only reported deaths there are for people who are immunocompromised who inhaled aspergillus into their lungs. And so the cannabis won't kill you, but what grows on it might if you're immunocompromised. And so they've mandated mold and fungus testing and bacteria testing in several states now. In fact, if you look at Colorado, they've mandated every 2 pounds be tested. You got to take a gram out of every pound or 2 pounds and put it through a microbiome type of test where you test for E. coli, salmonella, bacteria, and a variety of other yeast and molds. But they're doing about 240,000 pounds a year right now in Colorado alone, and it's growing 20% a year. So there's a large testing market in one state alone that's probably going to migrate to Washington and Oregon and Alaska and DC as those things become live. The other thing that's needed is sex testing in this field, because this plant has male and female plants. It's dioecious. So when it actually, when you plant the seed, you have a 50% chance it's going to be male or female. And the males, they aggressively get rid of. This is like Thelma and Louise on steroids, okay? They don't want males around at all because they pollinate the females, and the females end up stop making cannabinoids and they start making seeds. And so you can destroy an entire grow with a single male plant. Now, there are 13 federally licensed grow facilities in Canada that have licenses to grow 60,000 plants at a time. One male plant at 60,000 plants. Each of those plants are probably worth $1,000. It's a $60 million grow that a single plant can destroy. So they aggressively want to get rid of these, and the way they currently do this is they wait for them to grow 4 to 8 weeks, and they run around the place looking for them and rip them out by eye. Now, if we have the Y chromosome, we can figure this out the moment we have it propping out of the ground. So we're working on tests that can put all of this on a qPCR instrument. So the first ones that are coming out are yeast and mold, a salmonella test, and a coli test. They're a bit different. The yeast and mold, there's a tolerance of 10,000 CFUs per gram. The E. coli and salmonella are single CFU. And unfortunately, the regulations don't care if it's 0157. They just say any E. coli. So it's going to falsely fail a lot of material. There's coliform enterobacteria and then aspergillus. And the aspergillus one is probably very important because aflatoxin, in fact, requires CYP2C19 to be metabolized, and CBD turns that off, and it's a carcinogen. So they don't want to see any signs of this. And there are some games going on in the field where people will counterfeit traditional culture-based tests by just heating the material to 140 degrees and heat killing the bacteria. Okay, well, that might heat kill aspergillus. It's not going to heat kill the aflatoxin. And so the DNA sticks around through this, and this is one reason why we believe that PCR-based systems are a better approach. There's a couple of ELISAs you can use for these things, but it's a little bit, ELISAs can be dirty when you're grinding up plant matter and sticking antibodies to it. This is a very oily plant, so we're not very keen on the ELISAs. And then the other things in blue are XY testing, CBD and THC genetics, and some biome identification. We'll touch on a few of those briefly. I think you guys know why you'd use PCR instead of Petri dishes. I won't spend much time there. I think the important thing here to bring home, though, is that this is a case where a lack of a result equals safety. If there are no colonies on your plate, it equals safety. Okay, so that's an engineering flaw where you need to have a positive control. You need to know that whatever you did to put on the Petri dish worked, and that's much easier to do with PCR. You can put in internal controls to know, like, look, the reaction worked, I didn't miss pipette. The other thing that these labs aren't really used to is they're used to grinding up plant matter and running HPLCs. They're not used to amplifying anything. And microbiology amplifies your contaminant to measure it. It's just like PCR. So we're embedding a lot of the techniques that we've been working with NEB on to put methylated bases on these amplicons in qPCR so that, in fact, they're hydroxymethylated, so that we can chew them up with ABS1 and have very similar to a uracil decontamination system that makes material that we can then sequence. We haven't been going down the uracil road. Even though it's off patent, we haven't been able to cluster those things very well due to some polymerase limitations on the MySeq. So what we have instead of what you might find in the field is a biolumex system. This thing grows, and it measures in the tube a change in turbidity, and it measures a change in CO2, but it takes 48 hours for you to get answers, and we can do this in 20 cycles on a biorad. And we also have the benefit of having this internal control. That SCCG is a single copy cannabis gene. We sequenced 12 cannabis genomes to find regions that were in the cannabinoid synthase pathway that were fairly stable, which is not easy to do. There's a lot of polymorphisms in this plant. But we've targeted an amplicon that would be in every single assay, so we know the assay setup was right, and we have a positive control. And then we're targeting the ITS regions in yeast and mold. And there's a reason for wanting to target the ITS, which we'll touch on a little bit, but it's a nice microbiome tool in the end. And then there are these species-specific tests where we have to have a single CFU, and this is for E. coli and Salmonella, and we're just moving into enterobacteria, making sure that everything in that class we pick up with the same set of primers. These ones are a little bit more work because they're a broader class of microbes. So what folks can do if they get a positive result, the delta CTs can give you an estimate of the CFUs, but if you actually want to know what microbe it is, you actually want to sequence it. And that's important because most of the microbes in the plant are endophytes, like penicillium. These things are harmless, but they happen to grow in the plant, and if they're not there, you might get botrytis and other types of infections. So it's important to be able to not just enumerate, but to also identify. And so this is the reason why we selected the ITS region, and we tailed these with Illumina primers so that if people get a positive events, they can send back these by the dozen, and we can load them all up in a MySeq and get ourselves a nice microbiome report that might look like this. And what's really interesting here is what we're starting to see is we can identify who grew it by the microbiome fingerprint. This one has magnaporth in it. So this came out of Holland. We know exactly where it came in Holland. It came from DNA Genetics because they tend to always have magnaporth in the DNA they sent us. If you flip this around to greenhouse seeds, you'll see different organisms sitting in their grow room. So this has got folks interested in potentially looking at this for a surveillance system throughout the grow. You can imagine sampling 100 plants in here every 2 or 3 weeks, and this may seem asinine to do if you're just growing tomatoes, but if each of these plants is $1,000 and you have 60,000 of these under lights in Canada, running a couple plates of this every other week to understand, hey, is anything cropping up that we need to take care of? You can go rip the plant out and not have to put pesticides on it per se. One of the challenges they have in the testing market is there's like 200 pesticides that are not allowed and they have to test for. So a lot of these testing labs have some pretty expensive mass spec equipment to deal with the pesticide side of the testing. Folks that we've been talking to would rather just screen and rip them out than have to go and treat the entire room and deal with pesticide testing later. So it's opening up, we think, an interesting new field to bring some of these microbiome things into these very large-scale industrial grows. So when folks want to do large-scale industrial grows, they do not do crosses and plant them and rip out all the males. The reason they don't do that is because this is a real complicating problem. What they'll do is once they find a strain that they like, they'll go into a cloning operation, and they'll clone that plant. That way they don't have this problem with males popping up everywhere. If they ever want to make a new strain, like one that has high in CBD, because that's in high demand right now, they have to go through a breeding process and plant seeds and screen them, and that process can take 12 weeks. Now, they want to get the males out of there pretty quickly, and as I mentioned earlier, the way this was traditionally done was with an assay that was published by Mandolino back in 2004, but there's one single band here that tells you whether you're male or not. And that you can tell this is a pretty sloppy banding system to look at. I wouldn't want to be evaluating it if it had that many plants on the line staring at bands. And it's not very amenable to qPCR because every single one of them produces a product. So we cloned these products and sequenced them to figure out what was going on. And then we went and sequenced a couple males. I think an interesting hint that was in their paper was that this is a MAD-C2 repeat construct. So targeting a repeat as a tool, I have some problems with. It worries me that it's not going to be consistent. And from our estimates in sequencing a few male genomes, this is a couple hundred copies per chromosome, and there happens to be one particular set of SNPs on the male Y chromosome that makes this work. So you can see on the right side here, we have, first off, this is the 30x male genome, and when we align to this region, we get 17,000x. Now, granted, this was aligned with some loose criteria to try and understand what was going on, but you can see multiple different SNPs in the reverse primer. There's only one paired read across the whole region, and on the other end, there is a 3' SNP on that read. So it appears this is working because of one base on one primer to determine male or female. And we decided that what we'd rather do is take all of the female sequences that we have, take all the male sequences we have, align them against the females, everything that was left over and unaligned, assemble those into a putative set of Y contigs, and then design qPCR assays for those contigs, and graduate those into the laboratory and see what we can find. So this was pretty easy to do with just a Sage system and Nextera libraries. We just ran through this, and this was published as a technique in Lotus. They didn't have a reference for Lotus, so they compensated for that by having more plants, and they did a very clever K-mer approach here. But they were actually able to, with even some RNA-seq on top of this, nail the sex-determining locus, which is really interesting. Now, we can't be certain that we've nailed the sex-determining locus. We just know that we have a binary answer that's male-specific right now. And we've run this across 9 samples now that are male and about 20 that are female, and we haven't had any discordance yet. But it's a much cleaner answer than what we were seeing with the Mandolino set of primers, because those are very temperature sensitive, as you might imagine, and the gradients were giving us multiple bands. We've then converted this into a multiplex assay that again has our internal positive control, and you get a signal if there's a Y, and you don't get a signal if it's a female. And so this is ready to go out and it be expanded onto many more male samples. We've only got, as I mentioned earlier, 9 males and 20 females, but we think it's going to be a more reliable marker for qPCR for picking up X and Y. And we could potentially do that with a leaf cutting. Just a hole punch on a leaf will be able to get that type of information. Now, the other bit of information we think people are going to need at a very early age, at a seedling stage, is whether the plant's going to go down a route of generating THC or CBD. These genes are linked. They're in some type of linkage disequilibrium in the genome because they have found that you tend to get one of 3 genotypes with these plants. You tend to get things that make a lot of THC. They're a hybrid where they make 50-50 of THC or CBD, or they make all CBD. Now, the actual magnitude on that axis of how much CBD or THC they make, another publication has shown that a couple SNPs impact whether you end up being a 25% plant or a 12% plant. But this particular promoter region that Mandolino once again has described, we've gone about, when he described this, he didn't provide the sequences, he just provided some nice primers. We've then cloned those primers after they've been amplified and sequenced them to see what's going on, and we're starting to build assays that would pick this up as well, qPCR. So you can imagine getting a leaf punch very early on, knowing if it's going to be male, female, or CBD and THC just off of a multiplex qPCR assay. And we will probably go in later and make some SNP-specific assays as well to cover Sierra and Terpenes' work, where they showed that you can get very different amounts of THC synthase activity based on certain SNPs that are in the FAD binding domain of that enzyme. So there's a lot of genetics here that can be very helpful for breeders and marker-assisted selection. These are just some of these peaks from high CBD plants and high THC plants. We can actually clone these, sequence them, and design assays for them. And those are under design right now, so those should be probably available in the summertime. Now, the final thing, and one of the biggest things that's plaguing the field is that There is no trademarking in this field, and I don't place any judgment on there other than you can't go to the USPTO with a trademark, and people are renaming plant strains all the time. Many of you may have heard that the Marley family just announced that they're going to do a Marley natural strain of cannabis, and they're probably going to get 3 times the price of anything else, and whenever someone runs out of it, they're just going to rename that strain Marley Natural and sell it. And that happens all the time in the field, and the genetic screening that's been done in the field has shown that people are relabeling to whatever they can get the highest value to. So the chain of custody here can be solved with genetics. A lot of people have asked for DNA fingerprints. So where do you put DNA fingerprints of a plant that the trademark office won't accept? We've decided the best place is not necessarily NCBI for this, but to actually load these things into the Bitcoin blockchain. Because the Bitcoin blockchain instantly takes data and replicates it to 1 million different computers at once, and it's the largest supercomputer humming there is right now, and it has a financial motive to exist of a couple $2 billion of transactions going on in a day. So this is a very interesting play that we're thinking about doing. Well, we would take a RADseq library to get us maybe 100,000 SNPs off the genome. We would take those, we'd use those SNPs to draw a phylogenetic map, and also just as a sort of general fingerprint for that strain. And then if folks wanted to submit this with any meta information, we would help them submit an entry into a Bitcoin blockchain that would prove that this data exists at a particular time. Why would people want to do this? Well, if this is an easy approach to go, similar slide as doing RADseq. Why people want to do this is there is a motivation in the field for people to say, look, I just created a new strain. I want to publicly declare that it's mine, and I want to share its genetics so that anything that's ever shown up again, we can say whether it was derivative of that or not. And there's a bit of a first-to-file incentive here, perhaps from a bragging rights perspective, that, hey, we created a new strain, and we're the first to do it, and the first person who puts Marley Natural in here, every other strain that ever gets genotyped will get compared to that as the genesis block, and the phylogenetic trees will be based on whoever was first to file. So this is, we don't know, if you go to look into the blockchain currently, you won't see us submitting ATCs and Gs there. It doesn't accept data that way. It accepts a hash digest of that data, which is an encrypted form of communication, which is very interesting because it will be immortalized in an encrypted way, and the public and private keys can be turned either public or private. So people can submit data here, and only they can look at it, or they can choose to open this up for the entire world to use it. We don't own this database, so no one can buy us and shut it down. This database will be on public record and constantly holding a phylogenetic tree of all the species. And the reason for doing this, we believe is important to have it be a proof of existence and for it to be open source, because otherwise we don't think it's going to get used. We think if you are a private company claiming that you're going to own all the cannabis genetics, no one's going to send you anything. Frankly, there's too much distrust. So I think everyone wants to see it be a public record that everyone can compare their strains and their genetics, and we can know for certainty what are the genetics going around in the field. So we're hopeful it will pan out, but again, it's a bit of a gamble there. The main tool that is going out live already today, and we're installing this in MCR laboratories, is just a DNA prep that gets us 50 to 100 nanograms of DNA out of a very simple, quick prep with magnetic beads. And this can get us 10 PCR reactions or 2 genomes off of a prep. It's gauged to work with all of those mold testing techniques, but it's very field portable. You can do this on your kitchen counter if you had to, and it generates very nice Nextera libraries for sequencing, and we know it has absolutely no cannabinoids in it, so we believe it's safe to go in the mail. So there's a handful of folks that jumped on this as well. There's a few folks from the Solid team you might recall that have jumped on here and helped us get all these assays up and running. We've had a lot of helpful communication from MCR, Workshop, Tilray, and of course the people here at New England Biolabs have helped us design many of these assays for qPCR. And it's been a great collaboration because we've been able to pound through 4 or 5 different assays in 6 months, and it's all been working very well. And we've had some other esoteric forms of funding here. We've had a few reggae bands help us out, which has been really fun. They gave us free music and helped promote an iPad app that we got probably enough money off this iPad app to sequence a couple other genomes off of it. So it doesn't say much because genomes are so cheap to sequence these days. But and there have been some people throwing bitcoins around at us for some of the work, but it's all I think it's all more like a tipping jar than anything else. So with that, I'll just leave it for questions.
Could you go over rather slowly why male plants weeded out so vigorously?
Great play on words.
You went too
Yeah, so males don't generate any flowers, and flowers is the bud. That's what everyone wants for either consumption for recreation or for generating CBD. So if the male ever pollinates a female, the female quickly shifts from making flowers to making seeds. And so its cannabinoid content plummets, and then it starts to contaminate the flowers with seeds, and then people have to de-seed that low cannabinoid content flower. So they're petrified of having males because one male can pollinate an entire grow room, and suddenly a very valuable crop has now turned into something you have to throw away. So they aggressively have to go after these, and they do it visually currently. And the real challenge doing it visually is that the male sex organs can pop up in 24 hours. So if you're not paying close attention, they can pop up and start pollinating if you went away for a weekend. So they have to aggressively look at these things during a pretty broad window, 4 to 8 weeks. They're running around looking at all their plants to make sure none of them are male.
Okay, but nevertheless, you need some pollination to continue making seeds so that you can grow the next generation.
This is true, but once you have a plant that you like, you can clone about 100 of them in 10 days by clipping off a leaf and replanting it.
Oh, I see.
So when they find one they like, a female, they start cloning it. And now there's a hybrid vigor issue here. If they clone like 6 generations, they start to get stunted, and then they go back to doing crosses and finding females, and then repeating this. So the cloning process only takes you so many generations before they start to get stunted. Right in back.
Yeah. I've read, I don't know how true it is, but that along with the 2 active psychoactive ingredients, THC and CBD, that there are other psychoactive ingredients that modulate THC and CBD.
Yes.
Perhaps subtly, perhaps in other ways. But from what I read in the newspaper, the pharmaceuticals are always going for purified CBD and THC.
Yes.
Are they going to lose in all the other tail effects?
Yes, and that's a big debate in the field as to, the FDA-based model likes to isolate things into single drugs and test them. And when you start getting into, polypharmacy like this, the models fall apart. And there's a lot of literature on what you're talking about, which is called the Entourage Effect. If you have a plant that's high in myrcene, which is a terpene that comes through in some of these plants, that's an agonist for the CB1 receptor, and so it tends to potentiate the effect of THC. There's other terpenes that do the opposite as well. You'll find some in black peppers that will actually be an antagonist to CB1 and offset any of the high. So it is a complicated space on simplifying it. Now, the genetics for the other cannabinoids aren't as well understood. GW is probably the furthest on this, but as you mentioned, there's if you can recall the THC structure, there's a pentyl carbon string on the end of it. And there's propyl versions of this side chain that occur naturally in the plant that are called varins, or THCV or CBDV, and they have completely different pharmaceutical properties just by moving those 2 carbons. The plant makes everything also carboxylated, so it's only if you heat THCA does it turn into THC. So actually, if you take raw plants and squeeze all the THC out of it into an oil and give it to kids, they will not have a psychoactive effect unless you heat it. So those compounds have even different pharmacology. So this is a very interesting pharmaceutical repertoire. I mean, I liken it to people saying, look, this is like Pfizer going open source, over the counter tomorrow, because there's an enormous pharmaceutical repertoire in here that's soon going to be over the counter, and we need to tie this into personal genetics somehow, because there's too much potential to just have everyone grind this up as a bowl and smoke it. It's not going to go that way. For some of these patients, they need to basically take it as an oil to get the dosage right. You cannot give somebody 75 milligrams of THC without them probably jumping out a window. I mean, it's a tremendous amount of THC. You'll usually see personal doses in Colorado at 5 to 10 milligrams. And yet the epilepsy patients need it at that level, and they don't want it to be psychoactive. So they have to think about exactly what you're saying, is how do we combine this so it's not psychoactive so that patients can get a high enough dose. I think there's another question right over here.
I was going to ask about like hemp and agriculture.
I think there's going to be an enormous market there, and the males may matter there because they may grow stalk, and they may be easier to actually flesh out. And I think there's going to be marker-assisted selection activity there too to try and find completely different properties that make oil. In that case, you want seeds if you want to get oil out of the plant. There's probably other properties that we're not even looking at, and that's an enormous market. That's a market that, if it gets legalized, you can see the whole fiber and paper industry turning over to a hemp-based solution because it's apparently 4 times more efficient per acre.
For instance, if you on some epilepsy drug like Keppra, and if you consume for recreation purposes, can it interfere with the drug?
Okay, so it depends on the route of administration. If you consume it through smoking, it will bypass the liver, and you'll get a different effect. If you eat it orally, it will go through your liver and have an impact on some of... now, Keppra doesn't have a lot of drug-drug interactions, so we don't worry so much about that one. But what is important to know is when it goes through your liver, particularly THC, it gets 8 times more psychoactive. It gets converted into 11-hydroxy-THC. Which doesn't happen if you inhale it. It happens only if you eat it. So this is why some people have some very unexpected results when they eat it, is they don't gauge this correctly. They might have a grapefruit juice with it, and bang, it's much stronger than they anticipated. But other anti-epileptic drugs you do need to worry about. You need to worry about clobazam in particular and carbamazepine. Those two are interfering with the same pathway that metabolizes THC and CBD.
Psychosis and mood disorders. Has there been any analysis done to how these cannabinoids might work?
So there's a very active, it's a good point. So we haven't been getting patients of that flavor just based on, the way that we're selling our tests are into epileptologists who probably aren't handling psychiatric cases like schizophrenia. But there's a lot of data on Alzheimer's disease, Parkinson's disease, and schizophrenia having the endocannabinoid system play a very important role. There's a paper that came out just this week showing that low dosages of cannabidiol have a bigger impact on Alzheimer's disease and THC. Everyone was focusing on THC for Alzheimer's for a while, and this paper kind of changed that and suggested that the CB2 receptors are more pertinent. So for those who aren't familiar with the 2 dominant cannabinoid receptors that are out there, there's CB1, which is responsible for the euphoria and is mostly in the brain, and there's CB2 that's in your immune system. And the interesting thing about cannabidiol is it doesn't hit CB1, it does hit CB2, but it tends to limit neural inflammation. And neural inflammation has been really the focus of what it might be going on with CBD and epilepsy. That this is why some kids, very rare incidences when people get some vaccines, they get febrile seizures. It's because it boosts the immune system and they get a little bit more neural inflammation and they end up seeing seizures pop out. They think a lot of what's going on in Dravet syndrome is in fact a neural inflammation event, and that CBD is actually just dampening that down. So we think there's going to be a role in these other psychiatric diseases that may have a similar effect. If they're grounded in a neural inflammation, then it may play a role. There are clinical trials that are also going on studying Parkinson's and schizophrenia right now through GW Pharmaceuticals.
Do other plants make THC and CBD? Is it only the...
It's only been found in the cannabis plants, and there's one other contested one, a flower in, I think, South Africa that a group is looking at. It's not an identical compound. They just think it may actually, if you tritiate it, it may stick to a CB1 receptor. But no, they're fairly unique in their ability to make these compounds. What is common in the plant kingdom are terpenes. And this plant will make upwards to 100 different terpenes, usually 12 dominant ones that you'll find in most strains. But the terpenes are throughout almost the whole plant kingdom. You'll see there's probably 40 different ones in grapes. And the reason I connect that to the cannabinoids is that they are the precursor to making cannabinoid. It basically folds 2 terpenes together to make a cannabinoid.
I guess what I'm getting at is can the genes in the pathway of synthesis of these compounds be transferred over?
So that's the holy grail. People, there are companies starting to do that, try to take the artemisinin approach, where you clone the whole pathway into yeast. When they cloned a Japanese group cloned just THC synthase into yeast and tried to get it to express, and it got up to about 3 to 4% before it became an antiseptic and ended up killing the yeast. And so there might be some challenges in doing this because we think the plant may have evolved these things actually as a way of controlling its microbiome and being a pesticide. So they can be fairly cytotoxic to microbes, and they also believe that's what's happening in cancer. There have been some connections to these working in cancer because they are pro-apoptotic for cells that are growing very quickly. The mechanism of there is not completely understood, but there may be a challenge getting them through a synthetic biology route without solving that problem.
I'm not a chemist, but can these components be synthetically?
Yes, and it's a great question because they have, and they've been to market under the trade name Marinol. You have to be, they're often, when they're done synthetically, they're racemic. The plant is stereospecific, so there's a minor issue there. But as the other gentleman asked, people have not been reporting the same response rate when they take individual compounds. And Marinol was something that you swallowed, so people had this kind of erratic response based on their diet, I think, that they would get either overly dosed or underdosed because they couldn't judge getting it through their GI. So I think some of these other compounds in the plant may aid in managing that, or they'll find other routes of administration. They're seeing a lot of work being done transdermally right now. They're making like nicotine patches, except with THC or CBD in them, and those go venously through your skin, and it's a much smoother or a more predictable dosage path than going through your liver.
Okay.
All right. Great, thank you. It is.
Recognized her.
Oh, the initials.
Yeah.
Yeah, I should pull it out. He's consented, but
Well, I'm assuming so.
yeah, that's.
I assumed it wasn't you with the black belt.
Thanks. I'll just go use the soap now.